Hi everyone. Welcome to lecture 6, which is dedicated to the fundamentals of attention. Today we will be covering a number of topics, starting with a description of what it is broadly referred to nowadays as early and late selection theories of attention. We will continue with a discussion of two different types of networks, one called the attentional network and another one called the default network, and the relationship between the two of them. We will evaluate how a specific and relatively simple experimental paradigm has been used to study attentional modulation using a number of different neuroimaging techniques, including fMRI and EEG, and also a variety of population, including neurotypical, healthy adults of younger age and also of older age. We will look at how and why frontal lobe damage might increase distractibility. We will see how attention is needed to facilitate performance in a Stroop task. We will describe a family of mental tasks that appear to engage the same network, the so-called default network, and we will also consider the effects of degradation or dysfunction of the default network as related to a number of impairments and diseases. And last but not least, we will try to continue our more practically oriented discussion about how to read and how to assess an article, this time focusing on the introduction section of an article. Whenever attention is introduced in cognitive psychology or neuroscience classes, it is customary to start by acknowledging the contribution of William James, presumably the most famous American psychologist and also the author of the first introduction to psychology textbook. And William James made a number of contributions to the field, including detailing and explicating several basic intuitions about a number of cognitive processes. And one of these processes is attention. So in William James' own words, let's see what attention is. Well, everyone knows what attention is. It is the taking possession by the mind in clear and vivid form of one out of what seems several simultaneously possible objects or trains of thought. Focalization, concentration of consciousness are of its essence. It implies withdrawal from some things in order to deal effectively with others. Okay, so as William James points out, everyone has a basic intuition about what attention is. We talk about attention regularly, not necessarily in science, but in our everyday lives. And two things transpire from the way in which we talk about attention. One is the fact that it implies a limited capacity. We do not pay attention to everything that happens out there. We do not have the ability to pay attention to everything. And secondly, it implies selection. We select some things to pay attention to as opposed to others. Okay, so limited capacity and selection. Those are the things that William James drew attention to. We can place this in the context of a metaphor, right? We can think of attention as a spotlight on some things at the cost of others. If we try to be a bit more formal in terms of how we describe attention, perhaps a simple diagram might do. So let's say that the things that we can pay attention to are messages. They convey information about things that happen in the outside world, external stimuli, somebody's face, somebody's voice as we talk to them, or it can be something internal, right? Or what we reason what memories come to mind while having a conversation with somebody, or perhaps some distracting thoughts, my need for a cup of coffee while I'm engaged in a conversation with somebody else. So many things, both in terms of external sources and internal ones, can compete for our attention. And the idea is that we cannot really process all that information. Hence, we choose to selectively pay attention to a number of them. And we can describe this process of selection as a selective filter, something that information can pass through or not. Now, if something passes successfully through this filter, it can be further processed. And as a result of that processing, two things can happen. One is that we store that information in long-term memory, and the other one is that we may produce a response, let's say a behavioral response. We can answer somebody's questions or comments while being engaged in a conversation with them. We can choose to not pay attention to distracting thoughts or distracting outside stimuli. Okay, so this looks quite promising in terms of becoming more discrete, more clear about the way in which we typically refer to attention. However, this schema is also a bit too simple. So for instance, it doesn't tell us when and how that selective filter comes into play. If we talk about visual processing, for instance, you might imagine that that selective filter kicks into action at the level of early visual processing in the occipital cortex, or it might come into play while we're trying to recognize objects as performed and supported by higher level visual cortex, or even later at the point at which we are trying to identify the label or match a certain percept to previous experience stored in visual memory, right? So in that case, we have a diversity, a whole number of options in terms of when the selective filter is applied. And the second point that is not entirely clear in this schema, in this diagram, is how that selective filter works. Is it really strict so that some things pass through it and some don't? Or is it a bit more leaky, allowing some things to pass only to some degree and others not? Well, Donald Broadbent in the 50s came up with some cogent arguments that this selective filter acts early and it is very strict. So what Broadbent suggested is that it makes sense for attention to act as early as possible, since if you can eliminate information very early in the process, then you are simplifying and making a lot more efficient information processing during later stages. He also believed that the selective filter is quite strict. Information either passes through it or it does not. And if it does not, then it is lost forever. Okay, so now what is the evidence for this theory? Well, there's plenty of behavioral evidence, and we would definitely look into that if this was simply a cognitive psychology course. However, because it is a cognitive neuroscience course, let's look a bit at the neuroscientific evidence. And perhaps some of the earliest and more convincing, the one that shaped our thinking about attention, is due to EEG. Now, as you may remember from the earliest lectures, EEG is a method that exhibits considerable temporal resolution, but rather poor spatial resolution. However, due to its temporal resolution, this is particularly well suited to look into the timing of attention-based effects. So does attention act relatively soon or relatively late during, let's say, visual processing? So what do we expect to see when we collect EEG data? Well, as you may remember, EEG, and in this case we refer to scalp EEG, collects information about action potentials generated by a large number of neuronal cells at the surface of the skull. And if you study the shape of EEG signals, what you might see, especially in relationship with the occipital cortex, is something along these lines. So now, to remind you, whenever we look at EEG data, most of the time we average signals across a number of trials, since the EEG signal is intrinsically noisy. So we try to get better signal from EEG recordings by averaging dozens, if not hundreds of recordings corresponding to the different trials. And upon doing so and aligning those signals with, let's say, the onset of a stimulus, what we see are some recognizable deflections in a positive and negative direction relative to an average. Now, to avoid any confusion, if you look at this plot right here, you see that plus is the positive direction is plotted down and the negative one up. And this is a relatively old convention in the field, so nowadays some researchers still abide by it, or some completely discard it. But this is just a note that whenever you're looking at an EEG plot, you should pay attention to the y-axis and see where the positive and the negative directions are, whether they're plotted up and down or in the reverse direction. Okay, so we're looking at an ERP signal, an event-related potential, which is the result of averaging many EEG signals corresponding to many different trials. Let's say elicited by processing a visual stimulus. And what they capture are differences in the electrical signal. We're capturing electric potential. So what are some of the recognizable structures within such an EEG or ERP waveform? Well, if we look at it carefully, we see some relatively robust deflections, and we call this deflections components, or to be more precise, ERP components. Perhaps the most robust early component in response to a visual stimulus takes place around 100 milliseconds after the presentation of a stimulus, and it is conveniently labeled as P1. P because it is positive and 1 because it is the first one. Followed around 50 milliseconds later by another deflection in the opposite direction, a negative component conveniently labeled N1 because it is the first robust negative deflection of the waveform. Now, as I mentioned earlier, this is common for some electrodes placed over the occipital cortex. So in virtue of the fact that these components are early as 100 or 150 milliseconds after stimulus onset, and in virtue of the fact that they presumably capture primarily activation from the occipital or cortex, including the early visual cortex, but not limited to it, such components seem very good candidates for assessing the possibility that attention acts early rather than late. So let's be a bit more explicit and think about one way in which we can test this. Well, what we're looking at here is the possibility that attentional engagement might change some properties of these components. And this is exactly what a number of researchers did in the early 90s, including Ron Mangun. He also happens to be one of the authors of your textbook, along with Stephen Hilliard and Stephen Luck. Stephen Hilliard is one of the fathers of modern day EEG and its application in psychology. Well, Stephen Luck is the author of arguably the most famous textbook for EEG research in psychology. Okay, so what we've got here is a collaboration of researchers with a lot of expertise in the study of attention and a lot of expertise in EEG research. Presumably this work is quite compelling, right, in virtue of such expertise. And indeed it was compelling and also quite seminal. When they started this direction of research in the 90s, they looked at attention using extremely simple designs to simplify the reasoning and also the interpretation of their data. So in one such study published in 93, what they did was ask participants to look at the screen that contained simple visual stimuli that happened to flicker. So let's say we have 4 different visual stimuli displayed in different quadrants of the visual field, left, right, up, down. And then some of them flicker while the participant is paying attention to them or not. So for instance, you might be directing your attention to this stimulus that happens to flicker, or you might be directing your attention somewhere else while the stimulus flickers. So then using a procedure, or rather reasoning, that should remind you of the subtraction technique, what you can do is verify the difference in the structure of the ERP waveform as a result of whether the participant is paying attention to or not to that flickering stimulus. And what you see is that the amplitude of these ERP components changes quite a bit if the participant is paying attention to them. So if the participant is attending, let's say, the other side of the visual field, so let's say right instead of looking left, or looking at the bottom instead of looking up, what we see is a reduced amplitude in relation to what happens if the participant pays attention to that stimulus. And we see that this increase in amplitude happens at the level of P1, which is the earliest we might expect, as well as N1 and possibly also N2, another subsequent negative deflection in the ERP waveform. So over the next few years, these researchers try to get a better understanding of what exactly is going on. So there have been a number of follow-up studies that confirmed this observation and also provided a few more details. So for instance, a few years later, they looked into a different type of experimental design that had single stimuli flicker on one side of the screen or the other, in this case, round checkerboard stimuli. And what they found across a number of occipital electrodes, both on the same side with the visual field, called here IPSY, or on the other side, contra, deflections, or rather modulation in the amplitude of P1 and N1. So on both sides of the occipital cortex, we see that attention has a robust effect. It changes, presumably it amplifies the amount of neural processing that a stimulus receives as a result of attentional deployment. And this is indeed good news for Donald Broadbent's theory of early selection. So it seems that whenever you're looking at a visual stimulus, an attentional amplification of neural processing occurs early as related to early visual processing, early visual areas. However, to this stage, we only talked about vision. What about auditory processing? And it so happens that in the auditory domain, a very similar effect tends to take place. So, for instance, whenever you're presented with stimuli in both ears, you can pay attention to the stimulus presented to the right ear or to the stimulus presented to the left ear. So then what you can do is measure the difference in the amplitude of the EEG signal corresponding more or less to what happens at the presumed location of the early auditory cortex in response to such stimuli. And what it is typically found is the fact that when you pay attention to a stimulus presented in the right ear, the amplitude of the EEG signal is larger than the corresponding signal elicited by a stimulus which is not attended in the right ear. And conversely, we can find similar effects within the left ear. Okay, so we pay attention to a stimulus. There's a modulatory effect induced by attention early and is related to early visual processing and early auditory processing. And this is certainly good news for Broadbent's theory. It occurs that during the stream of perceptual processing, attention acts early, and it is quite likely in the sense that it does have a filtering effects. It changes the way in which we process information by devoting more resources to some things that we attend to. Yet there are some counters, some counterexamples to this theory of early selection, of filter theory. And some of these counterexamples come from everyday observation, not from scientific experiments. In case you do not remember the cocktail party effect, which you probably encountered in the Intro to Psychology class, this refers to a common experience that most of us have probably had. When we are at a party, at a gathering, we attend a conversation with somebody, we pay attention to that person, we engage in a conversation with that person, and suddenly we hear our name called from across the room. So somebody else in a different part of the room, having another conversation, mentions our name. According to filter theory, that information, that bit of perceptual evidence, perceptual message, should not make it through, should not become something that we are aware of, something that we become conscious of. It should be completely filtered out. Yet this is not what happens. Despite the fact that we are not paying attention by our own volition to that person on the other side of the room, when we hear our name called out or any other piece of information that might be quite relevant to us, let's say the name of a family member, or in case you happen to be a student in cognitive neuroscience, let's say somebody calling or mentioning something about cognitive neuroscience, that information does make it through eventually. So it seems that early filtering of information has its own limitations, and these limitations have been countered, have led to a theory developed by Anne Trisman in the 60s. It is called attenuation theory, in that it modifies Donald Broadbent's idea of a filter that completely discards information in favor of a more subtle and more complex approach when it comes to explaining attentional effects. To be more precise, what Anne Trisman suggested is that attention acts not just early, but also late. It's a multi-stage process. And secondly, she believed that attention is not a process of discarding information, but rather one of selectively attenuating information. While we pay attention to something by our own volition, some other information is processed less effectively. So let's say it is dampened or attenuated. But that doesn't necessarily mean that, especially in case when such information is relevant, it might not reach our consciousness. So we have an attenuation of information, of evidence, of perceptual evidence during an early stage, but during a late selection process, that information which has been dampened might still make it through to our consciousness, and in that way guide executive functions, decisions. It may get stored in memory, it may lead to a response, and so on and so forth. Okay, so this is attenuation theory. And it develops a bit more the idea of attention. It makes it a bit more complex. And it also leads to some interesting questions. Now, a lot of the evidence for this theory, especially in the early days, has been based on behavioral experiments. But there is a bit of evidence for it also from cognitive neuroscience. However, one of the issues that I want to take up in response to this theory is the cost of not paying attention to relevant information. And this is really important because if you believe Anne Trisman, if information is relevant, it doesn't really matter whether we pay attention to it or not. It eventually makes it through. It reaches our awareness, our consciousness. So then does it really matter whether we pay attention to it or not? And it seems that we do. So what happens if we pay or we do not pay attention to something which is relevant? And Mike Posner, one of the most influential researchers in the study of attention, designed a simple yet extremely effective paradigm which bears his own name, Posner's spatial cueing paradigm. What he did was design a very simple schema for an experiment in which a cue in the form of a arrow can point you in the direction of a subsequent stimulus or in the opposite direction. So what happens, to be more precise, in this type of experiment is that you see an arrow at the center of the screen, which can point you, let's say, in the right direction. And then after a brief delay, we can see a probe, a stimulus displayed in the right hemifield. And this is what happens on a majority of trials. So let's say on 80% of the trials, when you see a single arrow, the arrow points you in the right direction. And whenever the stimulus occurs, let's say on the right-hand side of the screen, then you press you press a button with the right hand, and when the arrow correctly cues you to the left hemifield and a subsequent stimulus appears over there, then you press a button with your left hand. However, crucially, on 20% of the trials, that cue is invalid. Which means that the arrow might point you in the right direction and the stimulus appears on the left, or the arrow points you to the left and the stimulus appears on the right. So we refer to that as an invalid trial. Now we also have a neutral condition in these experiments where the cue is not informative. So whenever you see something like two arrows pointing in different directions, you have no valid or invalid information with respect to where the stimulus might appear. So 50% of the time the stimulus might appear on the right and 50% on the left, but you have no advanced knowledge about where the stimulus might appear. Okay, so this is the paradigm. What are the results? Well, what Mike Posner showed, and many other researchers after him, is that there is a cost to invalid trials. Since valid trials occur on a majority of the situations, participants do indeed tend to attend to the hemifield that the arrow points towards. So then whenever the cue is invalid, there is a cost associated to redirecting attention to the opposite side of the screen. To be more precise, there is a cost in terms of reaction time. So we see here, for instance, that when the cue is valid, it takes participants less than 250 milliseconds to press a button correctly. While if the cue is invalid, it may take them about 300 milliseconds to make a button press correctly in response to the incorrectly or the invalidly cued target. And for neutral trials, we have something which is intermediary between those two different conditions. Okay, so it seems that despite the fact that we do attend relevant information, whether we pay attention to it or not, there is a cost to not deploying attention correctly to something. And that cost is quite substantial. If we are simply talking numeric values, that might not seem like much. It seems there is about a 70 to 80 millisecond cost in not deploying attention correctly. This is the cost of misguided attention. But if you talk about this in terms of relative numbers, this cost is about 1/3 of the reaction time for valid trials. And this is indeed meaningful. It is quite a robust effect. This is something that has been replicated many times in humans and other primates. Some of my colleagues even replicated this in fish. So it's a very robust effect, and it cuts across many species. It's fundamental in terms of how attention functions and in terms of the cost of not deploying attention correctly. Okay, so at this stage, we know that attention is a multi-stage process. We know that selection takes place both early and late. We know that there is a clear benefit in terms of attending to something, to a stimulus or to a task, in terms of speed and efficiency of processing. What we don't know yet is what is the neural architecture underlying attention. What are the brain regions that effectively support attention? And it only makes sense to ask that question in the context of a cognitive neuroscience class. There is no single region which is responsible for attention, and this should come as no surprise, since attention involves multiple stages of processing, and it also involves interactions with different sensory modalities. Instead, we have an entire configuration, an entire network of regions that span primarily the frontal lobe and the parietal lobe. We have regions such as the frontal eye fields in the frontal lobe and the intraparietal sulcus in the parietal lobe. And these are just regions that facilitate voluntary deployment of attention. So for instance, whenever I wish to pay attention to the slide in front of me, and I proceed accordingly. There is also an entire ventral network of regions that facilitate bottom-up attention when a loud noise occurs and suddenly my attention is drawn to it. However, that is not the contrast that I want to bring up here. Rather, I want to contrast the attentional network, this network of regions that supports directing attention and modulating the amount of attention to external stimuli and tasks, to a much more elusive network that has not been given sufficient focus until rather recently. And this is the default network, or the default state network. And it comprises, again, an entire number of regions that span of the frontal and the parietal lobe. It contains the medial prefrontal cortex and the posterior cingulate, and a large chunk of the lateral parietal cortex as well. And the role of this network is to facilitate what we often refer to as mind wandering, or daydreaming, or thinking to oneself. And that is something that we engage in extensively. Hence, there is that it's not a surprise that the brain allocates a specific network to it. What's interesting here is that our brains have a tendency to switch on and off between these two networks routinely. In more technical terms, these two networks are anti-correlated. We pay attention to something for a given amount of time, and then whether we want it or not, we disengage, and the default network kicks into play to facilitate or to allow what we refer to as daydreaming. Now, of course, we can re-engage attention when we want to. Ultimately, this is a network for voluntary attentional control. Yet the point still stands that we have two networks that are anti-correlated, that cover different aspects of our experience. One is dedicated toward focus to the outside world, to our interaction with a variety of sensory stimuli in the environment, while the other one can facilitate things such as remembering episodes in the past or envisioning the future, and so on and so forth. How might we go about identifying either one of those networks? Well, a very crude way of getting to, let's say, the attentional network is by using the subtraction method. We can ask someone to proceed with a very active attentional task, so perhaps completing a rather difficult task in response to a visual stimulus, and then taking a mental break while displaying the very same stimuli, for instance, to the participants. So again, we can subtract the activation that we find on the entire brain level when the participant is involved in the task. We can subtract from it the corresponding activation that we see when the participant is taking a brain, is becoming passive with in relationship to the stimuli in front of him. Okay, so we can use the subtraction method more or less to identify this network. And the story is a bit more complex in the sense that attention does not involve a single mechanism. If we talk about visual attention, for instance, that involves shifting attention to different locations in the environment many times. And whenever we shift our attention, we also have to move our eyes. So we process in a lot more visual detail things that we foveate, things that are covered, that fall on the center of our retina, on our fovea. So visual attention employs the control of eye movements, and it also controls in parallel shifting attention to specific spatial locations. But we may not necessarily pay attention just to locations. We might pay attention to different features of a stimulus, such as its motion or its color or its shape. So to that end, not only do different dorsal region come into play differentially, but also they interact differently to areas of the visual cortex, which might be responsible for motion processing, in this case V5, or color processing, in that case V4, and so on and so forth. So the story that emerges is that attention is a rather complex process, not just in time, but also in terms of neural architecture that allows it to function effectively. So, based on this understanding of the attentional network, how might one go about to verify, to check exactly how this entire network modulates activity in visual cortex, in visual areas? And first of all, whether this happens in the first place. Well, Ghazali and colleagues in 2005 came up with a rather simple yet very effective way to look into this using fMRI. So what they ask participants to do is view stimuli, either faces or scenes, in the context of 3 different tasks. One was simply passive viewing. We see alternating face and scene stimuli. And in the other 2 tasks, participants were asked to perform a specific task, to pay attention either only to faces or only to scenes. If they paid attention to faces, this was for the purpose of deciding whether they have seen a stimulus or not. So let's say participants see a face, then a scene, then a face again, then a scene, and then after a brief delay they need to make a response. So for instance, they need to decide whether new face they're seeing is one of the previous stimuli or not. And similarly, they can pay attention to scenes, and then they can make a decision a few seconds later whether the scene that is currently presented is one of the formerly viewed scenes. And the beauty of this design is that by and large it equates the demands of visual processing, in the sense that across these 3 different tasks, participants view the very same stimuli. The only thing that is different is what they do in response to the stimuli. Nothing, paying attention to faces or paying attention to scenes. So what are the results? Well, interestingly enough, if you look at the right PPA, this is the so-called parahippocampal place area, which is known to play a major role in the recognition of scenes. What they found was a clear difference in brain activity as a function of those 3 conditions. So what you see here on the y-axis is beta values. This is a specifically convenient, common way of measuring brain activity as a function of some rather simple regression analysis. And then what we see on the x-axis are the 3 different conditions. One happens when participants view scenes and they remember those scenes, they don't do anything, or they actively ignore them since they need to pay attention to faces. So with respect to the passive view condition, what was found is that activity is enhanced when participants pay attention to scenes within the right PPA. And this might not necessarily come as such a surprise, right? Since attention is supposed to enhance neural processing based on what we've seen previously with EEG. But perhaps what is more surprising is the fact that when participants pay attention to faces and actively ignore scenes, we also see a reduction of activity within the right PPA. And the more technical terms for that are enhancement and suppression. Brain activity is enhanced in the right PPA when participants pay attention to scenes, and activity is suppressed when they actively ignore scenes. The converse pattern of results is found in the right FFA, and this is good. It is certainly not just convenient, but it is reassuring that we see this with the other type of stimuli. So to be more precise, what we see is an enhancement of activity within the right FFA, which we know is critical for face recognition whenever participants remember faces, and a suppression of activity within the right FFA relative to passive viewing when participants ignore faces for the purpose of paying attention to scenes. Okay, so this at the group level seem relatively robust results. And sometimes one thing that researchers like to do is to see how consistent the results are across different participants. And Ghazali and colleagues did exactly this. They looked for this difference at the level of every single individual participant, and they found a surprising consistency. So while the amount of activity within the FFA might vary from one individual to another, and this is quite expected, the difference between the remember and ignore faces conditions tends to be relatively systematic. So we see this pretty much in every single individual out of 10 subjects. The difference can be reasonable, sometimes it can be barely observable, or it can be rather huge, as we see here. But what matters is the fact that in 10 out of 10 participants, we do notice a difference, and that occurs not just at the group level when we average across a whole number of participants. And this is quite impressive for any type of neuroimaging modality, this type of individual level consistency of a contrast. So this is what the fMRI data reveals. But Ghazali and colleagues did one other thing which is equally interesting. They looked not just at fMRI data within these participants, but also EEG data. And one of the most important components of the ERP signal when we talk about faces is the so-called N170, a negative deflection that occurs in response to face stimuli about 170 milliseconds after we view them. Now, whenever we try to assess N170, we can look at the amplitude of that deflection, and that is something that we already had the opportunity to discuss with respect to N1 and P1. But we can also look at the latency of a component. Now, it is true that N170 occurs more or less around 170 milliseconds after stimulus onset, but this timing is not really set in stone. There's quite a bit of variability around that estimate. And what Ghazali and colleagues found is that the estimate of occurrence of this component, N170, of this deflection, varied systematically with those two conditions. To be clear, whenever participants were asked to remember faces, N170 was systematically faster than when participants were asked to ignore faces, or rather to pay attention to scenes instead of faces. And you can see here the results of every single participant, and they match also the participants that we see here for fMRI data. And that is something that we very much like to see in neuroimaging. Consistency, not just across subjects, but also across different neuroimaging modalities. So in this case, fMRI data tells us that attention boosts activity within a specific region, which in this case is responsible for processing faces. But it also tells us, if we talk about EEG, that latency is adjusted. We speed up processing of things that are of interest to us. So this is a pretty compelling story about what attention does at the neural level and how it does it. It devotes more activity to a stimulus of interest, and it speeds up its processing. Okay, now all of this is quite interesting, and also the degree of inter-subject consistency is notable. However, there are things to keep in mind here. There are certain limitations. And one of these limitations is that, as it is often the case with neuroimaging studies or even behavioral studies, our population is quite homogeneous. These are all young adult individuals. So whenever we're looking at young subjects, this is what typically happens relative to passive viewing. Activity is enhanced, let's say, within the PPA whenever subjects remember scenes, and decreased whenever they're asked to ignore scenes. But what happened in older adults? Individuals of age 65 and older. And we're only talking about healthy adults, individuals who do not suffer from any notable or verifiable cognitive decrements or impairments. And the reason we're asking that question is because older adults often complain about attentional and memory problems. And those two things are quite related to each other. So what happens if we run the very same paradigm with older adults? Well, unsurprisingly, whenever they're asked to remember scenes, we see the advantage we previously noticed in younger adults as well, in terms of neural activation within the PPA. However, what might come as a surprise is the fact that the activation elicited by paying attention to faces at the cost of ignoring scenes is comparable to what we see in the passive view condition in the PPA. So based on these results, the story that this plots paint is that the problem is not so much with allocating attention to a specific type of stimulus, but rather the problem is with the ability to actively suppress activation within a cortical area when stimuli that we should be disregarded are not disregarded as much as they should be. So this is just one piece of a big puzzle with respect to how cognition changes during aging. And it is also one piece of the puzzle in trying to account for decrements in attention and memory, at least as related to visual processing. More intuitively, we see that older adults who complain about distractibility might have an explanation for the cause of that problem as a function of the inability of attention to filter out irrelevant information. So then a follow-up question is, okay, but what happens in patients? In patients who suffer damage to cortical areas responsible for the voluntary deployment of attention. And we have already seen that the frontal lobe, especially the medial frontal cortex, is critical for the deployment of attention. So what happens if we look at patients who have damaged, extensive frontal lobe damage? We do expect that they have problems with attention, but the question that Bob Knight and colleagues asked in '89 using EEG was what happens at the level of a visual cortex. So what happens if we record activity at the back of the brain in such individuals as opposed to normal healthy adults? And what he found foreshadowed Ghazali's findings who used fMRI about 15 years later or 16 years later. What Bob Knight found was that activity over the visual cortex was enhanced in response to certain stimuli in such individuals, in patients, relative to healthy controls matched for age. Why is this interesting? Why is this important? Because it suggests that attention has a critical role in not only selecting stimuli for beneficial or for more in-depth processing, but is also critical in that it leaves out, it filters out a whole number of distractors. And if attention does not work properly, essentially an entire range of stimuli, both wanted and unwanted, flood the visual cortex. And if we try to be a bit more formal about it, if we try to put together a schema of what's going on, you can think of the sensory input coming in, getting into the sensory memory, and then trying to make its way into the short-term memory. However, if attention does not work properly, then a lot of things are trying to make their way into the short-term memory, into the working memory. And that is something that we're going to touch upon a bit more in one of the future lectures. Now, one interesting question that emerges from all this research is related to automatic tasks. So far, we discussed about attention in the context of modulating or increasing the amount of neural processing dedicated to some external stimuli or some meaningful task in relationship with external stimuli. But many tasks are actually performed in the absence or in the apparent absence of attentional resources. Many tasks are automatic. For instance, many types of tasks that involve motor control, such as walking or even driving, may require little attention. And that is definitely not something that we would encourage, especially in the case of driving, but it is it does happen to a lot of us, and it is something that we would have to draw attention towards to be more careful. In the context of vision, another thing that we tend to process automatically in virtue of a vast amount of expertise with a task is reading. Most of us are experts at reading since our early childhood. Hence, we often tend to read without dedicating a lot of attention. So probably to most of us, this sounds like a familiar experience, just reading a piece of text, processing the orthography and the visual characteristics of the letters and of the words, but not really understanding what we have actually read because we have not paid attention to the meaning of those words and sentences. So reading tends to be automatic in the sense that the processing of the shape of the letters and the words is something that happens very quickly, very efficiently, and most of the time without awareness. And this is exactly what the Stroop task takes advantage of. So for those of you who do not remember or have not encountered the Stroop task in your introduction to psychology last year, this pins against each other two very different processes. One is reading and the other one is naming. To be more specific, reading reading words that denote colors, as opposed to naming the color of the fonts that make up a word. And what the Stroop task reveals is the fact that whenever you try to name the color of a word, that tends to be a slower, more laborious process as opposed to just reading the words denoting colors. So what happens in this case is that we're pinning against each other two opposite tasks. And one of them is automatic, hence it is naturally privileged. It is faster, it is more efficient, while the other one requires presumably more attention, and hence it can be slowed down by the attempt to suppress the automatic task. And there have been many studies using Stroop task over the last 2 decades using neuroimaging techniques. And this study by McDonald and colleagues from 2000 happens to be one of those landmark studies that tried to get to the involvement of the attentional network in processing words and naming colors in the context of a Stroop task. And what they found is quite interesting. So it should come as no surprise at this stage that areas of the frontal cortex would be involved in attentional control, in devoting specifically attention to the task in the context of a Stroop-based experiment. So in line with that expectation, McDonald and colleagues found that the dorsal lateral prefrontal cortex came into play much more so when participants were asked to name the color as opposed to reading the word. So whenever you're supposed to name the color of this word, red, as opposed to just reading the word, blue, it seems that DLPFC was a lot more active, was substantially more active across a number of scans. And this presumably happens because the DLPFC is responsible for allocating more resources when it senses that a conflict might be on its way, when it senses that more resources would be needed to adjust or to confront a higher level of difficulty for a task. Okay, so the DLPFC is in this case responsible for the allocation of attentional resources. However, when actually performing the task, it was not the DLPFC but a different region, the anterior cingulate cortex, that came into play. And this revealed higher levels of activation when participants were dealing with incongruent tasks or incongruent stimuli. So for instance, naming the color of words that refer to a different color as opposed to congruent stimuli when there was convergence in terms of the color of the word and what it denoted. And the interpretation of this finding is that the ACC is responsible for solving the actual conflict. So in this case, it seems that we have two different regions that are responsible, one for anticipating conflict and allocating attentional resources to deal with this future conflict. And then we have a separate region which is responsible for the actual management of a conflictual situation. And this is shown here in the context of a rather simple experiment relying on Stroop tasks, but this echoes many experiences that we have in our everyday life when we anticipate the presence of conflict or of difficulty, and hence we expect the DLPFC to play a meaningful role in the allocation of attentional resources. But then when we're actually dealing with a conflictual situation and we have to solve somehow the conflict in line, let's say, with our present goals and motivations and our assessment of different challenges and levels of risk, then it is the ACC that plays a critical role in that respect. So now that we have just passed the midpoint of the lecture, feel free to take a break, feel free to engage your default network, if you wish. Otherwise, let's continue our conversation with the default network. And we have previously mentioned the fact that in order to find the attentional network, one crude but effective way is to use the subtraction method to contrast region of the brains which are more active during an active task requiring paying attention to external stimuli, as opposed to simply taking a mental break. So you can imagine that we can also use the subtraction method in the opposite direction by contrasting activation when participants just take a mental break as opposed to an active attentional task that they're focused on. And you can think that this is relatively easy. All we would need to do is simply ask a participant to execute a task in response to some visual stimuli, and then we can proceed to display the same stimuli, but allow the participant to engage in mind wandering by simply not requesting anything rather than simply viewing those stimuli. However, this gets a bit more complicated because we don't really have control over mind wandering. This is something that is supposed to occur naturally. Hence, simply contrasting the levels of activation in response to an active task or just passive viewing might not be the best way to go about this. Fortunately, Christoph and colleagues in 2009 came up with a much better way, a more natural and more intuitive way to look into these issues. What they did, and this is quite common nowadays for this type of questions, is present participants with rather boring, uninteresting stimuli, which are likely to induce naturally a state of mind wandering in participants. So what they did then is intersperse the presentation of this stimuli with queries such as, was your attention focused just before this probe or not? And then the participants had to answer whether they were on task or they were off task, mind wandering over the last few seconds. So this is one question that participants were asked throughout the experiment. And where things get even more interesting is that that question was followed up by how aware were you whether your attention was focused? And the responses, of course, were aware or unaware. And this type of queries are examples of what is nowadays called experience sampling, in this case to study mind wandering. Okay, so then let's look at the results. What happens during mind wandering? Well, as expected, the default network tends to come into play. So we see some massive recruitment of neural resources from the frontal cortex, especially the medial part of the frontal lobe, but also areas of the parietal cortex coming into play. So this is not necessarily a surprise. What comes a bit of as a surprise is the fact that this is what we see when we mind wander without meta-awareness. When we mind wander but we don't realize that we are actually mind wandering. In contrast, when such meta-awareness exists, then we see a significant drop in activation at the level of these regions, both in the parietal lobe and in the frontal lobe. And this is in line with the idea that mind wandering, true mind wandering, occurs without meta-awareness. And it is in this particular case that the default network comes fully into play. Now, one thing that you might wonder about this is how exactly do we know that participants were indeed mind wandering? And this was verified both through self-report and by verifying their performance with respect to stimuli that have just been presented. For instance, we can ask the participant about some properties of the still images or of the video that you were just watching, and when they fail to respond correctly, then we have additional confirmation that mind wandering occurs. And again, the level of accuracy in those responses can also be correlated with the engagement, with the level of engagement of the default network as reflected by patterns of activation within the frontal and the parietal lobe. Yet, mind wandering is a relatively generic term, so many scientists try to get a better understanding of what type of specific things, specific thought processes falling under this generic term would engage the default network. And autobiographical memory is one of those things. Whenever you try to recall how you celebrated your 18th birthday, how you learn how to ride a bike, if you try to remember your first ever memory, all of that falls under the umbrella of autobiographical memory. And whenever we engage into processing autobiographical memories, we activate the default network. We also activate the default network whenever we try to envision the future. So let's say I'm trying to think about a coming game, and I'm trying to think about how the opposing team might react to a certain strategy, or I'm simply trying to think about how I'm going to plan the rest of the term, so I do my best at an exam. All of this falls under the umbrella of envisioning the future. And again, this activates the default network. Another example is provided by what we broadly call the theory of mind. And this refers to our ability to put ourselves into somebody else's shoes in terms of thinking and reasoning and feeling. So whenever I try to understand how somebody reasons in response to a particular stimulus, this reflects the theory of mind. And theory of mind is extensively studied in developmental psychology, because it's quite interesting the trajectory of being able to develop it. At our early age, we do not necessarily have a theory of mind. We simply project our desires and our goals and our reasoning onto other people, while later in life become keenly aware that people have other goals, other types of motivation, and consequently a different way of reacting, thinking, and feeling to external stimuli. So again, all of this falls under the umbrella of theory of mind. And whenever we try to think about what somebody else might be thinking, we engage the default network. To provide one final example, moral decision making also involves the default network. Whenever we try to pin against each other different decisions that find ground, let's say opposing or competing moral values, we engage this network. And why is this important? Well, because most of the time, whenever we talk about the neural basis of cognitive phenomena, one term that comes up over and over again is complex. We uncover more and more regions, we uncover more and more complex interactions, and so on and so forth. Well, this is one of those examples where things are actually becoming simpler and easier. If you happen to be a memory researcher, you would have thought a while back that these areas that span the frontal lobe and the parietal lobe are part of a memory system. And if you studied theory of mind, you would have thought that these are regions that are part of a theory of mind network. And if you studied decision making, you might have thought that these regions are part of a moral decision making network. When in reality, it appears that all of these networks are by and large the same one. They are actually the default network. And Randy Buckner is one researcher that made a significant contribution in this sense by outlining the similarities, if not the identity, of these different neural resources with respect to what nowadays we call the default network. And in recognition of the importance of the default network in a variety of aspects of cognition, its study simply took off, skyrocketed after the mid-2000s. So after 2006, 2007, we saw an explosion of neuroimaging studies dedicated to the default network. And nowadays we see hundreds of studies published every single year dedicated to more in-depth, more careful, more thorough descriptions of the default network and its relationship with other networks such as an executive network, an attentional network, and so on and so forth. So I would just note that it is quite ironic that we failed to pay attention to the default network until relatively recently, despite the fact that it is by and large one of the most actively used networks in our daily life when it comes to talking about neural resources. Until now, we have talked though about the default network in opposition to the attentional network. But is this always the case? Do we always switch one on so we can switch the other on or the other one off? Or is it possible that at times they work in cooperation with each other? And one interesting opportunity to study that is yielded by creative thought. Because if you think about it, creative thought definitely requires attention. But we also intuitively feel that creative thought can benefit from mind wandering. Some of our best ideas perhaps came to us when we are not particularly focused on a task. Or even if we are focused, it is perhaps when trying to envision the future that creative thought occurs. So there are arguments, intuitively at least, that creative thought might rely on both attention and mind wandering. And one study that looks specifically into that issue relied upon art students with demonstrably higher levels of creativity than most of us, for the purpose of studying creative thought in two different stages. So to be more specific, what they did in this study is ask art students to read a brief summary, a piece of text for 45 seconds. Then in response to that piece of text, they had to generate a drawing, right? So they were directly asked to be creative in terms of generating a drawing. Then they proceeded in a controlled condition to just trace a contour of an image, and this is just for the purpose of isolating areas responsible for motor control as opposed to creativity. However, what happens more interestingly is next in an evaluation stage, when the very same participants were asked to look at what they generated and think about what went well, what was not so well, what can be corrected, and so on and so forth. So then what happens if we look at the cortical areas that support generation of visual art as opposed to evaluation of the drawings that these participants perform while while being in the scanner. Well, if we look at the contrast between generation versus evaluation, a small but significant number of regions come into play, and most of them belong to what we would refer to as the attentional network. And this is not necessarily a surprise. Whenever you generate, you have to be very careful, you have to be very focused, and that requires attention. More interestingly, though, when the contrast between evaluation and generation was performed, when we look at brain areas that are more active when evaluating than generating on drawing, then not only did areas of the attentional network come into play, but also areas of the default network. So for instance, we can see some of the familiar culprits by now, such as the dorsolateral prefrontal cortex. We can see the dorsal anterior cingulate region. We can see parts of the parietal lobe, including the temporoparietal junction and the precuneus and so on and so forth. So a whole lot of regions come into play, and it doesn't really matter their precise identity at this stage. What matters is the fact that surprisingly, it seems that at least in this context, both the attentional network and the default network are recruited in the process of evaluation. And this might seem surprising at first, but it might also be the key to understanding creativity. So a particularly intriguing hypothesis is that creativity is facilitated by more flexibility in how the attentional network interacts with the default network, and the ability of these two networks to function in parallel and in cooperation, as opposed to being in opposition all the time. It also raises a whole lot of other interesting questions. For instance, whether this also underlies creativity in other artistic domains. So let's say in music, or in literature, or in non-artistic domains. So for instance, we can talk about creativity also in science and in business. Is it again because of the interaction and the flexibility in how these two networks work with each other that some individuals are more creative than others, and also the same individuals are more creative at some times as opposed to some other times? So these are some very interesting questions to the field. Now, before we conclude the theoretical part of our discussion today, I do want to touch upon the role of the default network in a number of diseases and types of cognitive impairment. And I'm going to start with Alzheimer's disease because in all likelihood, many of us know somebody who suffers from Alzheimer's. Because of the increasing incidence, increasing prevalence of Alzheimer's disease within the population, the forecast by the Society for Neuroscience is by 2050, most, if not all of us, will have known someone who suffers from it. And as you know, this is a neurodegenerative disease with extensive complications in terms of and impairments in terms of spatial abilities, memory, language, and so on and so forth. So there's been a tremendous amount of resources and energy spent trying to understand what's the neurological basis of Alzheimer's disease. And one thing that we had the opportunity to discuss earlier during this term was amyloid deposition. The role of beta amyloid plaques in neuronal death and the atrophy of certain cortical regions as revealed by PET. So now we can touch upon a more sensitive issue, and that is where exactly is this deposition taking place. And more recently, it was found that there is a surprising localization of amyloid deposition with respect to the default network. So to be more clear about it, we see a prevalence of beta amyloid plaques within regions circumscribed by what we call the default network. And there are a number of hypotheses as to why this might happen. One which is particularly interesting is the fact that the default network is particularly active from a metabolic standpoint. We involve in mind wandering pretty much all the time. Whether we think about the future or we engage in a social interaction, and hence we have to involve regions that support the theory of mind, or whether we try to retrieve something from our own memory. So in all of these cases, the default network tends to come into play, and that increased metabolic cost seems to be associated or to facilitate amyloid deposition in those regions. Hence, as a result of that, we see higher levels of atrophy at the level of those cortical regions. The plaques form, they disrupt the connectivity between neurons, they disrupt the ability, the access to resources for those neurons. And as a result of that, we see large numbers of cortical areas being compromised. And hence, since these areas are part of the default network, it is not a surprise that we see impairments in memory, in envisioning the future, in moral decision making, in theory of mind. All of those types of cognitive phenomena that rely heavily on the default network. Now, this is not by far the only theory or the only neurobiological basis of Alzheimer's disease, but it's certainly a step forward. And the work conducted by Randy Buckner and his colleagues certainly opened a whole lot of paths of research. We cannot begin to treat a condition before we have a good understanding of what is its neural basis, what parts of the brain are impacted, why, and how. And this is exactly what this specific piece of work has tried to do here. Okay, so let's move on to a very different condition now. Let's talk about a bit about schizophrenia. And its relationship with the default network. If you are a student in clinical psych, then you already know that schizophrenia is commonly associated with hallucinations, perceptual experience that are not based in reality, as well as with irrational thought, with delusions, lines of thought that most of us would consider to be unrealistic. There's been a great deal of research trying to understand what is the neural basis of schizophrenia. And in one such study, Whitfield, Gabrieli, and colleagues looked at the default network in controls in neurologically healthy adults as contrasted with patients who have undergone a clinical diagnosis for schizophrenia, as well as the first-degree relatives of those patients. So that would mean mothers or fathers, brothers or sisters of these individuals. And what they try to do here is quantify and localize activity within the default network in controls, in contrast to relatives of patients and the patients themselves. And what they found at first was that by and large the same regions of the default network came into play across these different groups of individuals. However, critically, they found overall more activation within the default network, and overall ampler, a wider recruitment of large swaths of cortex within the dorsal and frontal lobes in patients and their relatives with respect to controls. So based on this, it appears that part of the answer to understanding schizophrenia is a hyperactivity of regions of the default network. Now, the story gets even more interesting in that if you look at how tightly related these regions are, regions of the default network, in patients who suffer from schizophrenia, you can find a significant positive correlation between the severity of the disease, the psychopathology, in the level of connectivity between areas of the default network. So not only are these areas hyperactive, but they also tend to be hyper connected. And this provides some strong evidence that the default network is certainly involved in schizophrenia. Whether it is the cause of it or not is a lot more difficult to tell, since again, what we're looking at here is correlations, and to repeat the familiar adage, correlation does not imply causation. But it certainly opens up a lot of interesting paths of research into schizophrenia. And one other thing that I should mention here is the fact that a more in-depth evaluation of these results also revealed less anti-correlation between regions of the attentional network, such as the dorsolateral prefrontal cortex, and areas of the default network, such as the medial frontal cortex. So it seems that sometimes the cooperation between those two regions can be related to disease. And this is something to keep in mind, because previously we mentioned that the cooperation between these two regions seemed to be key to creativity. Yet in some other circumstances, we can see that a lack of anti-correlation between these two networks can lead to some severe impairments. So this paints a much more complex picture of the relationship of these two networks, the attentional network and the default network, their function, their utility, and our ability to interpret what goes on in the level of neural architecture with respect to these networks. Okay, that concludes the theoretical part of the lecture for today. And we're going to move on into the practical part, which concerns skill building, reading the intro part of a journal article, of an experimental article. So the same thing that we did for abstracts last week is something that we can do for the intro, for the introduction part of an article. So let's look at some of the things that an introduction tends to do or should do. And one of those things is to contextualize the particular area of research. In simpler terms, why do the authors do what they do? What's the fundamental motivation? And to a large extent, that motivation is theoretical in nature, but many times it is also practical. So many authors try to emphasize the real-world applications and relevance for a specific field of study and for a particular experiment, for instance. Okay, so this is mostly an explanation of why they do what they do, and they try to provide a bigger picture before going into details. Secondly, they try to clarify what we know about a given area of research and what we still don't know. What areas are well understood and what aspects of the field are rather unclear or controversial. Third, they try to articulate how a given study can get to what we still need to know. What kind of experimental paradigm is it used? What type of analysis? What type of population will be investigated? So all of these things need to be motivated in terms of their ability to get to a specific question. And fourth, many times authors try to provide an informed hypothesis regarding what they expect the results to look like. So they often use terms like we hypothesize, or we predict, or we expect, or we anticipate, and so on and so forth. And this happens a lot, either explicitly or implicitly, because much of the research in neuroscience, and not just in neuroscience, but in pretty much every field domain of empirical research, is hypothesis driven. So we make some hypothesis, and we expect the empirical evidence also to provide confirmation for them, or to falsify, to provide arguments and evidence that our hypotheses are incorrect. Conveniently, when all four elements noted above are present, they tend to occur in that order. However, that doesn't necessarily mean that they all need to be present, or they need to be present to the same extent. So, for instance, there are many exploratory studies that complement hypothesis-driven studies. So when we approach a new field of research, for instance, we might not have much intuition or make any intelligent guesses about what we might find as a result of some structured observation or analysis of a large set of data. And it's important to have both types of studies, both exploration and hypothesis-driven research. But of course, when exploration is involved, then clear hypothesis, well-motivated hypothesis might be missing from the introduction. Also, it's becoming more and more common nowadays to have studies that combine those two elements. So you might have one or two hypotheses and some area of research or some aspect of research which is left over for exploration. Okay, so then how do you understand an introduction? How do you know that you have a proper and sufficient understanding of the introduction section of an article? Well, the same strategy that was applied for understanding an abstract can also be applied here. I would recommend trying to summarize the points made in an introduction. An introduction is a bit longer than an abstract, hence it can be very useful to try to summarize it within 3 or 4 points. However, if you expect some challenges in forming a good impression of an introduction, even using this strategy, you'd be correct. So here are some of the most common challenges that even educated readers face when processing the text of an introduction. By far the most common one is the appeal to new and complex concepts that you have no familiar with in advance. And related to that, there may also be reference to experimental protocols that you haven't encountered yet. So whenever that happens, you have to decide whether a concept and a protocol seem fundamental to what is being discussed and used in a study. Then perhaps this is time to do a bit of a Google search. Or if you happen to be in a position of a textbook, then you can also look through that textbook. Try to gain, in a nutshell, a quick understanding of what those concepts and those protocols refer to. Third, there may be references to other studies that you haven't read before. So whenever somebody else's work is mentioned that you haven't encountered yet, then that can pose a challenge. So again, if that work is critical, you might need to do a quick search. Otherwise, you can try to infer just by looking through the text what that work might have done and what might have contributed to research in the past. And sometimes introductions or later the discussion sections of an article might try to make that a bit clear to help the reader. Fourth, introductions can sometimes be quite long, and that would be for good reason, because they try to delve into a rather complex and extensively researched field of research, but that can also make it difficult for you to keep your focus, to keep your thread while going through it. Now, articles, especially for neuroscience, tend to have manageable length introductions, and the higher the impact of a journal, the shorter an introduction tends to be. However, if you read introductions from articles in reputed journals dedicated to cognitive psychology, such as Psychological Review, then you might encounter multiple page introductions that do extensive reviews of prior work in the field, rather than quickly delving into the topic of research in a specific study. So do keep in mind that depending on the profile of a journal and the type of research being discussed, an introduction can range from anywhere between one single paragraph or many paragraphs across multiple pages. Okay, so then what better way to develop this skills, introduction reading skills, than to look at a couple of papers and see how effectively we can parse and we can understand what is presented to us in the introduction section. And we're going to quickly look at two papers. One of them is a paper that I co-authored 2 years back along with a number of researchers from Carnegie Mellon University. And this paper looked at recovery after surgical resection of parts of the occipital temporal lobes. But let's try to go through this more strategically, more systematically, and see how can we navigate an introduction. So one of the main suggestions would be to do this one paragraph at a time. And the reason to do that is because many times the introduction section is quite constrained. So neuroscience articles, as I mentioned, the higher the impact, the more restrictive in terms of the amount of information and the number of words you can put in an introduction. Hence, as an author of an article, you have to be extremely careful and judicious in terms of what information you place in the introduction and how you structure it. Hence, the paragraph structure of an article can resonate quite well with the logic of a study and with the anatomy that I have just mentioned, the anatomy of an introduction. Okay, so this is a introduction for a paper published in Cell Reports, and the intro is quite short. It only consists in 2 paragraphs. So let's start with the first one. And whenever you're reading a paragraph, especially if you are new to this process, to reading scientific articles, I would suggest trying to synthesize the ideas from a paragraph and put together some quick notes. This is what I used to do when I first started to read papers, and it really helps. As you develop more and more skills with reading scientific articles, you might be able to discard that. But even nowadays, whenever I feel that an introduction is particularly stuffy and difficult to comprehend, I try to break it down and jot down a few notes that summarize the content of different paragraphs and of an intro. Okay, so then let's look at the first paragraph of this introduction. And we're going to go through this sentence by sentence and see what are the things that we should react to and how. Okay, The human ventral visual Watt pathway projecting through the occipital pole to the anterior temporal lobe comprises a host of category selective areas. Okay, so if you read this sentence, that's good news for us. We understand that the object of study is the ventral visual Watt pathway, which we have already covered in previous lectures. So we already have quite a bit of background information of what this study addresses. This topography follows a well-defined medial to lateral arrangement in adults and is highly reproducible across individuals. Okay, so these are also relatively well-established ideas that we have encountered. There is a topography to how the ventral Watt pathway aids object recognition, and we also know that there is a systematic structure which is reproducible across individuals. So nothing truly new up to this stage. It's just setting up the study. Whether this cortical organization is fixed or alternatively plastic is unclear. So here you can see how hypotheses might start to sneak in. So we know that we know a lot about cortical visual organization, but we don't know the extent to which it is plastic. It can adjust to different constraints. Examining the impact of surgical resection, e.g. lobectomy, and possible restitution of function can therefore offer unique insights into the reorganization of the cortical visual system and the mechanisms of functional recovery after brain injury. Okay, so here is where things might start to become even more interesting. So we understand that the way in which the authors have looked into the plasticity of visual cortex is by examining probably patients who have undergone lobectomy. So this might be the best time to go online and find out what lobectomy is, and also try to find some pictures since presumably this is a surgical procedure. So some scans, some structural scans might be able to show you exactly what a lobectomy looks like. And you can also look through the paper in question to find something like this. So a lobectomy is defined as a surgical removal of a lobe. So this is not the result of an accident or of a disease. This is something that a neurosurgeon will do. They will remove a considerable amount of brain structures, including gray and white matter. So either the total or the almost total removal of a lobe in one hemisphere or the other. And then if you look at the pictures here, you can see exactly what happened for one particular patient. So you can contrast the shape of the brain and the structural integrity of the brain pre-operatively and post-operatively. So you can see that after the surgery, there's a huge chunk of occipital temporal lobe missing. Okay, so this is important because lobectomy is one key term that we need to understand before moving further. But this sentence is also useful because it introduces us to some context, right? It tries to motivate why this is interesting. It is interesting because it provides unique insights into the mechanisms for functional recovery after brain injury. Okay, so there's not a whole lot of detail about context here, but we expect that recovery after brain injury is something that a lot of people care about, and with good reason. So it might not be necessary to go into a lot more detail as to why we need to look into mechanisms that facilitate recovery. Okay, moving on. To date, little attention has been paid to the recovery of function following the resection of regions of the cortical visual system. Okay, theoretically, one may postulate a continuum of possible outcomes ranging from no plasticity to complete reorganization. Okay, so here come into play the hypotheses. We have two opposite hypotheses pinned against each other. One is no plasticity, the other one is complete reorganization. Moving on, the extreme case of no reorganization is predicted by the hypothesis that the functional topographic map in the ventral cortex is innately pre-specified and that individual regions have a single assigned function which may be immutable. Okay, so this defines or brings more clarity to what no plasticity refers to. We already talked about functional specificity. So if functional specificity is innate, if you're born with it, and if it doesn't tolerate any change, then that essentially implies the lack of reorganization of visual cortex following surgery or disease or accidents. Okay, so this is one hypothesis. What about the other? The opposite extreme is the view that all brain areas are equipotent, which predicts that many, perhaps even all regions may be plausibly assume the function of another region and recovery ought to be complete. Okay, so the extreme hypothesis, the other end of the spectrum, is that let's say if the FFA is damaged or completely removed, given to its location in the occipitotemporal cortex, then another region which has been left over either on the other hemisphere or the remaining parts of the same hemisphere should be able to embrace that functionality of the FFA. Okay, of course, there are many other patterns of structure functions correspondences that fall between these endpoints. So here you can sense that the authors try to play it safe. They pin two extreme hypotheses against each other, but it seems more likely that the truth of the matter lies in between those two hypotheses rather than at the ends of the spectrum. So then, based on what we have discussed so far, we can look into what the 3 points from this paragraph might be. Well, one is that the ventral pathway hosts category selective areas, and we already talked previously about things such as the FFA and the PPA, so this is not new. Lobectomy may surgically remove these areas, and again, this is not necessarily a surprise. So some of you might be surprised that neurosurgeon is allowed to remove such a huge part of the brain, but of course, if you remove such a huge part of the brain, then it should come as no surprise that the entire FFA might be eliminated. And then that leads us to the main question of the study. How much reorganization occurs afterward? So once you remove the FFA and the PPA and a whole bunch of other category selective areas, what happens with the rest of the brain? Do we get other brain areas which previously played no role in face processing to suddenly become face selective? Well, let's take a look at the next, the second and last paragraph of this introduction. To assess the nature and extent of reorganization of the residual visual cortex, we track the longitudinal changes in cortical organization in a pediatric case following surgical removal of the right ventral occipitotemporal cortex as a result of pharmacologically intractable epilepsy. Okay, so there's a lot of information here. First of all, there's an inkling into why such a massive removal of brain structure was performed. This was because of pharmacologically intractable epilepsy. So we have patients suffering from epilepsy, and no amount of type of medication was able to deal with that problem, and presumably the severity of the condition is so high. So we are left with a neurosurgery approach as a last means of treatment. The other things that transpires from this is that we're looking into a pediatric case, so we know suddenly that we're dealing with children. It is confirmed that the type of lobectomy concerns the right ventral occipitotemporal cortex, which was also shown in the picture on the previous slide, and that we are tracking the reorganization of the visual cortex over time. This is the meaning of longitudinal changes. It's not something that you do just one, you do multiple times across an entire span of months or even years to find out exactly how reorganization takes place. Pre-surgically, UD showed typical activation in the retinotopic cortex of both hemispheres, and language was left lateralized. Okay, this is good to know, and this is something quite common in the neuropsychological literature. You want to establish that your patient was fine prior to an accident or prior to a surgical procedure, because otherwise you don't know whether the impairment that occurs later is due to the actual procedure or to some pre-existing condition. Okay. Let's see next. Post-surgically, UD had a persistent left homonymous hemianopia as confirmed by 2 visual perimetry tests conducted 3 years apart. Okay, so here's some terms that we might have a bit of familiarity, but not completely. We have encountered the talk of about hemianopia in our previous lectures. We know by now that this is the loss of vision with respect to one visual field, one visual hemifield, either left or right. So what is homonymous hemianopia? So if you do a quick search, you'll find that this is the loss of one hemifield on the same side for both eyes. So what that means is neither eye can provide you with information about either the left or the right visual field. And this presumably happens because the occipital lobe, or one hemisphere, is compromised. It also introduces the idea of visual perimetry tests. So if you're curious about it, you can go through the paper or you can go online and find a picture that shows you what perimetry test results might look like. And it is something like this. It's showing you what parts of the visual field are compromised by displaying crosses. So crosses in red show parts of the visual field where the participants cannot tell you, for instance, whenever a line is shined, right? And green shows you parts of the visual field where vision is perfect or close to perfect. And then you can also have yellow, which indicates part of the visual field which might be slightly compromised. And if you think about this patient had damage to the right occipital lobe, so it only makes sure, based on what we know about visual processing, that the left visual field is impacted, and that no eye can effectively present us with information that is efficiently informing us about what happens in the left visual field. Okay, so moving on, starting at 13 months post-surgery, age 7 years, 10 months, and during the subsequent 36 months, age 10 years and 10 months, UD was scanned 5 times and changes in functional selectivity and or topography of the higher order and early visual cortices were explored. Okay, so this is also letting us know a bit about what was being done. And while not mentioning specifically fMRI, it mentions scanning. And scanning is often a keyword or a cue that what was done here was a form of scanning that relies on MRI. So presumably fMRI was conducted, probably some structural MRI as well. And it is also telling us that the focus of the investigation was on early visual cortex and higher order visual cortex. Okay, the last sentence tells us that psychophysics experiments were conducted in parallel to characterize any changes in visual behavior. Okay, so we have fMRI investigations which are coupled with behavioral testing within the same child, referred to here as UD. So now let's try to figure out what the key points of this paragraph are. Well, we know that there's typical visual and language processing pre-surgery. We know that post-surgery we have left field hemianopia. And then we know that the authors are interested in finding out how and how much early and high-level visual cortex change post-surgically over time. So this also allows some amount of exploratory research. We know that there are some big hypotheses driving this research, but the authors like to play it safe. And they're asking not necessarily which hypothesis is correct out of the two outlined on the previous slide, but rather how much change, how much reorganization do we see in between those two extremes. Okay, so this is an example of one relatively brief introduction. Let's move on and talk about another intro for a very different article. This is one article authored by one of your DAs, Tyler, and published last year. And what I'm going to do is let you read through the first three paragraphs of this introduction, and then I'm going to try and articulate the main points of each paragraph one by one, and I will ask you to see whether they match more or less your own thinking about what kind of information they present and they're trying to convey. So I'm going to pause for a few seconds, and then I will proceed through each of these paragraphs one by one. Okay, so assuming that you managed to go through this, let's see what the first paragraph tells us. Well, we find out that there is a concept that people refer to as summary representations, and this is probably key to the research conducted in this study. We understand that a summary representation is a mechanism involved by perceptual processing to deal with the wealth of information, the wealth of perceptual information by summarizing it, by averaging it into something convenient. We also find a bit about the context, about the bigger picture, which is presented in the first two sentences. We often look at groups or crowds of faces in many real-world circumstances. So a lot of the research is focused on single faces in neuroscience. In real world, we often have to provide the perceptual analysis of many faces perceived or witnessed at the same time. Okay, so we have a context and you have a main concept, summary representation. Okay, then let's move on to the second paragraph. Well, this seems to provide us with a bit of background. There is a bit of research coming from fMRI that tells us where summary representations might be processed, and also a bit of EEG research that draws attention to things like the N170 component and its sensitivity to processing groups of faces. However, perhaps the main point of this paragraph is conveyed by the first sentence. And this motivates the current study. It is telling us that there's been plenty of behavioral work on the topic of facial summary representations, but there is relatively little work from cognitive neuroscience using neuroimaging methods. Hence, this is a gap in the field that we feel compelled to fill. Okay, let's move on to the third paragraph. And if you go through this, it's telling you the methods that will be used. So pattern analysis and image reconstruction methodology, which was introduced and discussed last week, apply to EEG data. So we know the how. Now the question is, what are we looking for? And the first sentence of the paragraphs provides us with some hypothesis that single faces, as opposed to crowds of faces, may exhibit, when processed, different neural profiles. And since we're looking at EEG research, then presumably what this entails is that if you look at the waveform corresponding to someone looking at a single face versus looking at a whole bunch of different faces, may look quite different. So one waveform in response to just seeing a single face, and a very different waveform when looking at crowds of faces. However, again, you see that there might be some room left over here for exploration, because the second sentence here tells us that we don't really have any clear idea about what is the visual content of summary representations. So people in the field have talked about what a summary representation is, but we haven't been able to visualize it before. We don't really know how that looks like in the mind's eye, so to speak. And presumably this is exactly what image reconstruction does. It explores the visual content, the appearance of a summary representation. What is it? What does it look like? Can we visualize it in the first place? Okay, so these are some of the things that should come to mind, and you can try to pull out of the three paragraphs of such an introduction. Okay, so then I'm gonna leave you with another intro, or rather just the first three paragraphs of an introduction that belongs to an article published by another one of your TAs. I'm trying to be fair, so I can present some of the work of different TAs in this course on different occasions. And if the text here is not clear, or if you want to take a better look at the content of this article, I will also make available via Quarkus an electronic copy. But one of the questions in the practice questions for today, you will be asked to summarize the main points of the first three paragraphs of this introduction. Okay, so good luck with the practice questions for this week, and I look forward to continuing our conversation next week on a very different topic. We will proceed with a discussion of action.